Chronic myeloid leukemia, or CML, is a clonal hematopoietic malignancy characterized by the Philadelphia chromosome, a genetic abnormality that arises from a reciprocal translocation between the long arms of chromosomes 9 and 22 to produce the chimeric BCR-ABL1 oncogene. The BCR-ABL1 oncogene encodes a tyrosine kinase, which binds ATP and becomes constitutively active. The constitutively activated BCR-ABL1 tyrosine kinase leads to an increased CML cell proliferation and resistance to apoptosis, which results in an increase in myeloid cells or leukemia. Tyrosine kinase inhibitors, or TKIs, work by occupying the adenosine triphosphate, or ATP binding pocket, of the BCR-ABL1 kinase domain. This binding stops the enzyme signaling and results in the death of target cells. Despite this success of TKIs in the treatment of CML, BCR-ABL1 positive cells can be present in patients who achieve a complete cytogenetic remission. These cells potentially form a reservoir for future resistance and disease progression. Although resistance is believed to be multifactorial and includes both BCR-ABL1 independent and BCR-ABL1 dependent mechanisms of resistance, point mutations within the BCR-ABL1 kinase domain are a major cause of resistance and treatment failure. Indeed, many amino acid mutations have been described, with some occurring at higher frequencies than others. Topographical changes within the binding pocket that sterically hinder TKI binding, or changes to amino acid side chains, may result in direct disruption of binding between a TKI and the BCR-ABL1 protein. A variety of mutations occur on various amino acids. Different amino acid substitutions can also occur at the same residue. Based on published literature, including the NCCN and ELN-CML guidelines, 21 clinically relevant mutations have been identified. The threonine 315 to isoleucine mutation, or T315I, is one of the most frequently reported mutations in clinical practice. The insertion of a bulky isoleucine prevents the formation of a crucial hydrogen bond between a TKI and BCR-ABL1, and this sterically hinders TKI binding. The T315I mutation is resistant to all first-generation and second-generation TKIs. This mutation has been associated with disease progression and poor survival. Ponatinib was designed using a computational chemistry and structure-based drug design to inhibit BCR-ABL1, including drug-resistant mutations that arise during treatment. Ponatinib, a third-generation BCR-ABL1 TKI, is a unique, rationally designed molecule with structural features that allow it to display the profile of a pan-BCR-ABL1 inhibitor. Ponatinib was designed using a computational structure-based approach to incorporate two key design elements. First, the molecule has a carbon-carbon triple bond linkage that allows key interactions with the side chain of the T315I gatekeeper mutant. Second, the molecule has an optimal fit to the BCR-ABL1 binding site, which allows it to be effective against a broad range of mutants. These features allow Ponatinib to confer high potency against native and mutant BCR-ABL1, underlying its pan-BCR-ABL1 inhibitor profile. Ponatinib inhibited the in vitro tyrosine kinase activity of BCR-ABL1 and T315I mutant BCR-ABL1 with IC50 concentrations of 0.4 and 2.0 nanomolar respectively. Ponatinib inhibited the in vitro viability of cells expressing native or mutant BCR-ABL1, including T315I. In preclinical studies, Ponatinib has shown activity against 21 clinically resistant mutations, including the most resistant T315I mutation. Ponatinib is a pan-BCR-ABL1 tyrosine kinase inhibitor that decreases the growth and increases apoptosis in CML cells.